Imagine a single breast cancer cell.
Right.
It's born in the breast tissue, right? Just this microscopic rogue agent, and it somehow manages to break free. It navigates the incredibly chaotic, rushing currents of the bloodstream.
Which is pretty hostile territory for a rogue cell.
Exactly. It survives that journey, which in itself is, frankly, statistically improbable. But then it arrives at the body's ultimate security system, the blood-brain barrier.
Right, the BBB.
And it hacks through. And once inside the brain parenchyma, it does something that, honestly, it basically borders on science fiction. It suddenly learns to speak the chemical language of neurons.
It really is the ultimate infiltration. I mean, the pathology here isn't just about a mass taking up physical space.
Right.
The tumor actively disguises itself as the very neural tissue it's invading.
Yeah.
It integrates into the local network.
When a breast cancer cell pulls this off, it feels like dark magic. But a groundbreaking 2026 study in the journal Cell, Death, and Disease just turned the lights on for us.
It really did.
They didn't just look at bulk tumors to see what was happening. They literally caught the cancer cells in the absolute act of learning this new language. So our mission today for this deep dive is to pull apart exactly how breast cancer brain metastasis, or BCBM, occurs. We're going to explain every mechanism, every detail.
And the level of resolution they achieved here, that's really what makes this study a landmark. They use something called single-cell RNA sequencing, or cRNA-seq.
Right.
They analyzed a massive amount of data, 131,880 individual cells from 7 different patients.
Wow.
Yeah, and they sampled everything along the metastasis route, the primary blood supply, the cerebrospinal fluid, the adjacent non-tumor brain tissue, and of course the brain tumors
So
themselves.
it's incredibly comprehensive.
Very. And they reinforced all of that with bulk DNA sequencing from 47 additional patients, just to catch those broader genetic mutations.
Okay, let's unpack this, because the methodology itself is just fascinating. I mean, single-cell RNA sequencing fundamentally changes how we look at tissue.
It does.
To use a food analogy, taking a biopsy and doing traditional bulk RNA sequencing, it's like throwing a bunch of fruit into a blender, taking a sip of the smoothie, and, trying to guess the exact ratio of strawberries to blueberries just by the taste.
That's a great way to put it. You just get a blended average.
Right. But cRNA-seq, that is like freezing that smoothie in time, suspending it in midair, and examining every single individual strawberry seed and blueberry skin to see its exact molecular state.
Yeah, that distinction is vital. By pulling that cellular smoothie apart, the researchers didn't just see generic, cancer cells and immune cells.
Right.
They saw the active transcriptomes, essentially the real-time state of mind of every single cell at the exact moment it was captured.
Which brings us to the actual battleground inside the brain, the tumor immune microenvironment, or time.
Right, the time.
If we look at the immune side of this environment, the brain has its own local resident police force, but the tumor also brings in external mercenaries.
Exactly. So the local police force consists of microglia. These are the resident macrophages of the central nervous system. They basically live in your brain full time.
And how did the researchers spot them in the data?
Well, in the cRNA-seq data, they identified them by specific protein markers on their surface. Think of these as their police badges.
Okay, badges.
For microglia, you look for TMEM119, P2RY12, and SAIL1. Under normal conditions, their job is to patrol the brain parenchyma, clear out debris, and eliminate threats.
But the tumor doesn't just deal with the local microglia, right? It actively recruits external immune cells from the bloodstream.
Right, the mercenaries.
The tumor-associated macrophages, or TAMs, and they carry different badges entirely.
They do. They specifically express markers like LYZ and CD163.
Now wait, if I'm a cancer cell, bringing in more immune cells sounds like a terrible survival strategy. I mean, why import your own executioners?
Because macrophages are highly plastic. They can exist in different functional states, which we generally categorize under the M1 versus M2 paradigm.
Okay, M1 and M2.
Right. M1-like macrophages are your classic fighters. They are pro-inflammatory, they release cytokines that destroy pathogens, and they aggressively attack tumor cells.
But M2?
M2-like macrophages, on the other hand, are the builders and the healers. Their evolutionary purpose is to suppress inflammation and promote tissue repair, usually after an infection is cleared.
Oh, I see where this is going. If an immune cell switches into a healing and building mode while inside a tumor, it's not healing the patient.
No, I don't.
It's building a nice, cozy, vascularized home for the cancer.
Exactly the problem.
Yeah.
And this is the massive discovery the researchers made when analyzing the single-cell data. The circulating TAMs, the mercenaries found in the blood, and the cerebrospinal fluid, they were almost entirely M1-like.
So they were in fighter mode.
Yes,
Yeah.
heavily expressing markers like LST1 and cell. But the TAMs that had actually physically crossed into the brain tissue and become resident inside the tumor,
Let me guess, they flipped.
they had completely flipped. They were now M2-like, expressing markers like APOE, SPP1, and most notably NLRP3.
Okay, I want to make sure I have this straight before we move on. Let's do a quick recap for you listening. Number one, single-cell sequencing lets us look at this tumor ecosystem one cell at a time.
Right.
Number two, the brain has local cops, the microglia, and blood-imported mercenaries, the TAMs.
Exactly.
And number three, incredibly, those TAMs flip from an attacking pro-inflammatory state in the blood to a tumor-protecting healing state once they get inside the brain. So think about your own immune system for a second. If you were a cancer cell, why waste energy fighting the immune system when you can just bribe it to act as your personal bodyguard?
It is brilliantly insidious. But, it immediately poses a massive biological puzzle. How do we actually know they are flipping?
Right, because if we are just taking a static snapshot of the brain tumor, how do the researchers know these M2 macrophages used to be M1? Couldn't they just be two entirely different populations of cells that, just happen to be in the same place?
That is where the computational biology gets truly impressive. They used an algorithm called pseudo-time analysis.
Pseudotime.
Yeah, combined with a technique called RNA velocity.
How does that work?
Well, RNA velocity looks at the ratio of spliced to unspliced messenger RNA inside a single cell.
Okay, unspliced meaning it's brand new.
Exactly. Unspliced RNA is newly transcribed straight from the DNA. Spliced RNA is mature and ready to be used. So by mathematically comparing the two, you can actually predict the future state of that cell.
Oh, wow. So it acts like a developmental time machine.
Precisely. It allows us to draw a precise evolutionary trajectory from one cell state to the next.
So they didn't just guess. They traced the exact developmental path of these mercenary TAMs.
They did. And the trajectory shows they start as cell-positive TAMs in the early stage, circulating in the blood.
Okay.
And as they transition, they evolve into PRKCB-positive TAMs in the middle stage. And finally, in the late stages inside the brain, they mature into LST1, APOE, and SPP1 positive TAMs.
The full M2 builders.
Right. But the absolute terminal final stage of this evolution, the most mature, heavily M2-like mercenary in the brain, is the NLRP3 positive TAM.
The final boss of the tumor macrophages.
The final boss, yeah. And this NLRP3 TAM doesn't just sit there. It actively brainwashes the local cops, the microglia.
How? How does a cell brainwash another cell?
To prove this, the researchers used an analytical tool called cell chat, which maps out cell-to-cell communication.
Okay, cell chat.
It essentially looks for cells pumping out specific chemical ligands and matches them to cells expressing the receptors for those ligands.
Like a locking key?
Exactly. And the cell chat analysis revealed that these terminal NLRP3 TAMs act as massive signal broadcasting hubs. They secrete incredibly powerful immunosuppressive molecules.
Which ones?
Specifically, transforming growth factor beta, or TGF-beta, and interleukin-10, or IL-10.
Oh, wow. I know TGF-beta is a heavy hitter in the immune system. When the mercenaries pump out TGF-beta and IL-10, what does that actually do to the local microglia receiving the signal?
It completely suppresses their anti-tumor immunity. TGF-beta directly inhibits the production of inflammatory cytokines. It suppresses the activation of cytotoxic T cells.
So it forces the microglia to just drop their weapon?
Literally. It reprograms their internal transcription machinery, so they also adopt an M2-like tumor-protecting state.
Okay, let's do another quick recap. To summarize this twisted communication network for you. Number one, the TAMs evolve through a specific developmental timeline, ending up as terminal NLRP3 positive macrophages. Number two, these terminal TAMs act as localized bosses, secreting TGF-beta and IL-10.
Yes.
And number three, this chemical signaling acts as a biological brainwashing agent on the local microglia, forcing them to stand down and protect the tumor. So knowing that tumors can literally reprogram our own defense mechanisms to act as their bodyguards, how does that change the way you view cancer treatment?
It's terrifying, right?
Completely. You aren't just fighting rogue cancer cells anymore. You're fighting a corrupted version of your own immune system.
Which fundamentally changes the therapeutic approach. You can't just poison the tumor, you have to break the communication lines.
Right.
But to understand how to do that, we really have to look closely at the cancer cells themselves.
Right, to see what they are actually doing with this perfectly safe, immunosuppressed nest they've built.
Exactly.
Let's talk about the invaders, the metastatic tumor cells, or MTCs. Now, one of the things that stood out to me in the data was the stark difference in homogeneity versus heterogeneity.
It is a striking contrast, because if you look at multiple brain metastases across different sites within the same patient, they share a very high degree of genetic homogeneity.
They're practically clones of a successful invader.
Right. But if you compare the invading MTCs between different patients, there's massive transcriptional heterogeneity. The cancer cells are utilizing their DNA blueprints in wildly different ways depending on the person.
Even with all that interpatient chaos, though, the single-cell RNA sequencing allowed researchers to group these invading MTCs into distinct molecular subtypes, right?
Yes, 5 of them.
Yeah.
MTC blank, neuro-related AR plus HR plus AR plus 80, and HER2 plus 80.
Okay, 5 subtypes.
But the HER2 positive subtype requires special attention here. When they tracked the cellular populations moving from the blood, across the barrier into the brain parenchyma, and then into the cerebrospinal fluid, the proportion of HE2 positive MTCs steadily increased.
So they were thriving.
Exactly. This gives us clinical evidence that HE2 positive cells possess an exceptionally high intrinsic capacity to breach the blood-brain barrier. They really are the master infiltrators.
But how do they actually pull off the invasion? I mean, a breast cancer cell shouldn't physically be able to thrive in the brain. The study breaks their colonization down into what we can essentially view as a 3-stage bank heist, right? H-E-I-S-T.
A heist is a perfect analogy for the pathology here. Stage I is the break-in. In this early stage, the MTCs show intensely high active energy metabolism alongside massive upregulation in transcription and translation processes.
So they're burning through energy reserves.
Yes, to physically and enzymatically pry open the tight junctions of the blood-brain barrier.
Okay, so that's the break-in. Then comes stage 2, the disguise or the camouflage. This corresponds directly to that neuro-related subtype we just mentioned.
Right.
Once they are inside the brain parenchyma, these breast cancer cells activate biological signals for synaptogenesis and neural signaling.
And just consider the biological implications of that for a moment. A cell derived from mammary gland epithelium is suddenly expressing the genes required to build a neural synapse.
It's bonkers.
What's fascinating here is they survive the harsh alien environment of the brain by mimicking brain cells.
They learn the local language to avoid detection and access local resources. And then finally, stage 3 is the base camp. Once they are established, the MTCs downregulate that high-energy state and start producing cell adhesion properties.
Right, they drop the heavy camouflage and focus on physically sticking together.
Forming the dense physical tumor masses we actually see on an MRI. So, recap time. Number 1, invading tumor cells are categorized into 5 subtypes, with HER2 plus cells leading the charge across the barrier. Number 2, they execute this invasion in a 3-stage heist high energy for the break-in, neural camouflage to mimic local brain cells, and adhesion to form the structural tumor mass.
Perfect summary.
So here's a question for you listening. Does the idea of breast cancer using neural camouflage to disguise itself as your own brain cells fascinate you, or does it terrify you? Honestly, for me, it's a terrifying testament to cellular adaptability.
It is clinically daunting, for sure, because the immediate glaring question is, how does a breast cancer cell even access that part of the genetic code?
Right.
Where does it get the blueprint to suddenly synthesize neural receptors?
This brings us perfectly to the genetic keys. We need to talk about the specific mutations driving this mimicry, starting with copy number variations, or CNVs.
So a copy number variation is a type of structural mutation where large chunks of the genome are duplicated or deleted. The researchers found that these specific brain metastatic cells had massive amplifications. Basic extra copies of chromosomes 8, 16, and 21.
And a deletion too, right?
Yes, a significant deletion on chromosome 13. This widespread genomic instability is the chaotic foundation that allows the cancer cell to start playing with genes it usually ignores.
And within that chromosomal chaos, they identified 2 primary culprits, the genes NRXN1 and GABRB3. Now, NRXN1 usually encodes NRXN1, which is the synaptic adhesion molecule. It's literally the biological Velcro that holds two neurons together at a synapse.
Right.
And GABRB3 encodes a subunit of the GABA-A receptor, which is a ligand-gated ion channel that neurons use to receive inhibitory neurotransmitters. Again, just to be absolutely clear, a breast cancer cell has absolutely no business expressing a neurotransmitter receptor.
None at all. It shouldn't happen.
Yeah.
But here is the most counterintuitive and arguably the most crucial mechanistic detail of the entire study.
Okay.
When the researchers sequenced NRXN1 and GABRB3 in these tumors, they weren't finding mutations that altered the protein structure itself.
Wait, really?
Yeah, there were no missense or nonsense mutations changing the shape of the receptors.
Yep.
Almost all the mutations were non-coding regulatory variants.
Non-coding.
Yeah, specifically located in the 3' untranslated region, the 3' UTR,
Right.
or in the intronic regions of the genes.
I want to use an analogy here because non-coding mutations can be tricky to wrap your head around.
Sure.
Imagine a gene is a song. A normal coding mutation would be like someone changing the actual lyrics or the melody of the song. It changes the fundamental structure of the final product.
Right.
But a non-coding regulatory mutation, that's like leaving the lyrics perfectly intact, but breaking into the soundboard and jamming the master volume slider all the way up to maximum. The song's exactly the same, but it's deafening.
That perfectly captures the mechanism of action. These non-coding mutations in the 3' UTR disrupt how the RNA is degraded or regulated. They don't change the shape of the NRXN1 or GABA-A receptors.
So what happens?
They just cause the RNA expression of those genes to massively, unstoppably increase. The cancer cells are suddenly flooded with structurally perfect, highly functional neural receptors.
And this brings us to the ultimate terrifying connection that ties this whole ecosystem together. We have to bring back the immune system. Remember those terminal NLRP3 TAMs?
Yeah.
The corrupted mercenary bosses pumping out signals?
Right, the brainwashers.
Huh.
The ligand receptor analysis didn't just show them brainwashing microglia with TGF-beta. It showed those NLRP3 TAMs are also pumping out potent growth factors.
Specifically insulin-like growth factor 1, or IGF-1.
Exactly.
And who is perfectly positioned to catch that IGF-1? The camouflaged metastatic tumor cells. Because they've cranked up the volume on those neural receptors like NRXN1 and GBRB3, they utilize them as massive satellite dishes.
It's wild.
The corrupted immune cells send out the growth signals, and the camouflaged cancer cells receive them, fueling their own aggressive expansion. It is a closed, deadly feedback loop of survival.
It's an entirely self-sustaining ecosystem. The tumor doesn't just survive the hostile environment of the brain. It actively engineers the local immune cells to feed it.
So just to recap the genetic side of this nightmare. Number 1, brain metastases rely on heavy mutation burden, specifically copy number variations.
Right.
Number 2, the mutations in the NRXN1 and GABRB3 genes are mostly non-coding. They act like volume knobs, drastically turning up the production of normal neural proteins to achieve camouflage.
Yes.
And number 3, the corrupted TAMs in these mutant cancer cells use these receptors to create a feedback loop that continually feeds the tumor. So here's a thought for you. If these non-coding mutations act like volume knobs, could future targeted therapies be designed to just turn the volume back down on those specific RNA transcripts rather than relying on, systemic toxins to violently kill the cell?
That is precisely the therapeutic avenue this research opens up.
Wow.
But, a massive single-cell sequencing data set is still essentially correlative. The researchers had to prove causality. They had to take this to the lab for in vivo validation.
Right, because computer models are great, but how do we know this isn't just mathematical noise?
Exactly. So they started in a petri dish with highly aggressive human breast cancer cell lines, specifically MDA, MB231, and MCF7.
Okay.
They utilized small interfering RNA, or siRNA, to effectively silence the expression of the GABRB3 and NRXN1 genes in these cancer cells.
They manually turned the volume knob back to 0. What happened?
The cancer's aggressive phenotype basically collapsed. Without those neural receptors, cell proliferation, their ability to migrate, and their capacity to invade extracellular matrices just plummeted.
But a petri dish is a highly controlled environment. It's not a living, breathing brain with a functioning immune system.
Which is exactly why they escalated to an in vivo mouse model.
Ah, okay.
They took a mouse breast cancer cells, called 4T1-LUC, and used CRISPR-Cas9 to stably knock out the GDR3 and NRXN1 genes.
They just edited them right out.
Yep. They then injected these genetically edited cancer cells directly into the left cardiac ventricle of BLBC mice.
Right, the heart. Why the heart?
Because this administration route, pumps the cancer cells directly into the arterial circulation, sending them racing straight up to the blood-brain barrier.
Oh, this is the ultimate test of the heist mechanism. Can they still break in and set up base camp without their neural disguise?
Precisely. And the researchers monitored the metastasis using IVIS bioluminescence imaging,
Which allows you to visually track the glowing cancer cells inside a living mouse, right?
Yes. And they confirmed the findings with post-mortem H&E tissue staining.
And the results?
The results, undeniable. The mice that received the cancer cells missing GABRB3 or NRXN1 developed significantly fewer brain metastases.
Wow.
And even more importantly for clinical relevance, those mice demonstrated significantly prolonged overall survival compared to the control group. Without the capacity for neural mimicry, the cancer simply couldn't colonize the brain tissue effectively.
And they didn't stop at mice either. To close the translational loop entirely, they cross-referenced this with actual human clinical pathology.
They did.
They performed immunohistochemical staining on real physical tissue samples from human breast cancer patients. And the physical evidence proved the sequencing data. The protein expression of these neural genes was highly elevated in the patient's brain metastasis tissues when compared back to their original primary breast tumors.
Which means it's physically happening in human beings exactly as the single-cell algorithms predicted.
It is a rigorously constructed chain of evidence. They move from computational pseudotime algorithms to in vitro human cell lines to living mammalian models and finally validated it in human clinical pathology.
It's an airtight case.
So final recap for our listener. Number one, silencing these neural genes stops breast cancer cell growth and invasion in a petri dish. Number two, deleting these genes in mice prevents brain metastasis and genuinely extends survival. And number three, clinical tissue staining proves this volume knob effect is actively driving metastasis in real patients. So given how incredibly effective deleting these genes was in stopping the spread in mice, how quickly do you think the pharmaceutical industry can turn this into a viable, targeted therapy for humans?
Well, translating a systemic gene silencing therapy from mice to humans is incredibly complex, particularly when you're dealing with the blood-brain barrier.
Sure.
But identifying the precise genetic volume knobs, knowing exactly which wires to cut, is the absolute non-negotiable first step. We couldn't target it before because we literally didn't know the tumor was speaking this language.
Which really brings us to the profound reality of this entire deep dive. We started by asking how a simple breast cancer cell manages to hack the ultimate security system of the brain. And by pulling apart the tumor smoothie, single cell by single cell, this study mapped out a terrifying yet scientifically awe-inspiring ecosystem. We found blood-borne mercenaries corrupted and brainwashed into acting as tumor bodyguards.
Yep.
We found cancer cells executing a highly coordinated 3-stage heist, exploiting non-coding mutations to literally speak the chemical language of neurons, all to create a feedback loop that ensures their survival.
It really forces a paradigm shift in oncology. Cancer is not merely a disease of unchecked rogue cellular division. It is a disease of dark, sophisticated adaptation. It learns a local environment, it manipulates intercellular communication, and it ruthlessly exploits our own repair mechanisms.
Oh, yeah.
But because technologies like single-cell sequencing allow us to intercept these communications, we finally know exactly how the tumor is orchestrating the bribe.
And that leaves us with one final, deeply unsettling, yet kind of mind-expanding question to chew on. We've just spent this time exploring how breast cancer cells can adapt to the brain by learning to mimic neurons and hijack localized immune communication. But what happens when cancer spreads somewhere else? What other localized languages are tumors learning when they metastasize to the liver or the bones or the lungs? Are they learning to speak the chemical language of a hepatocyte or an osteoblast to survive there too?
It's a chilling thought, but biologically it's highly probable. Uncovering those organ-specific dialects is undoubtedly the next great frontier in metastasis research.
Thank you for joining us on this deep dive into the cellular underworld. We hope this changed how you view the microscopic battles happening inside the human body. Keep questioning the science around you, keep looking for the intricate mechanisms behind the mystery, and we'll see you next time.